This is the 10th video in the Fundamentals of Biology, and today we're talking about Mendelian genetics. So Mendel has two laws of heredity. The first one is called the law of segregation, which means only one of two alleles is passed on to sex cells. And that means that alleles don't blend together. So in this diagram, each parent will only donate one of their letters or alleles. So this one will give a capital R and this one will give a lowercase r. On the other hand, the law of independent assortment means that alleles separate independently of other alleles. So in this case, the characteristic of being yellow or green does not affect whether or not it is wrinkled. Here are some basic terminology in genetics. Genes are DNA that code for a specific trait and are passed down from parents. So genes are found within chromosomes in this first picture. And the loci is the location of a specific gene. So in this case, the trait coding for eye color is found here, whereas hair color would be found in a separate location. An allele is basically a version of a gene, and usually a trait will have two versions possible. One version would be the dominant allele, and this is denoted with a capital letter. On the other hand, our recessive allele is denoted with a lowercase letter. When an offspring is homozygous, it means an offspring has received two of the same allele from each parent. So in this diagram, the first and second pictures are homozygous because they have the same letters. However, if they have different letters and receive different alleles from each parent, it is called heterozygous. The genotype is the inherited alleles that the individual has. So this first picture shows the genotype of certain offspring. However, a phenotype is what a trait actually will show. So in this last picture, you can see a dark rabbit, another dark rabbit, and a white rabbit, given different genotypes of two big G's, big G, and two little g's. So a monohybrid cross will determine the probability that an offspring will have a certain genotype. And in this case, we're going to be crossbreeding two heterozygous parents for a certain tall trait, with a capital T being tall and a lowercase t being short. So the first step is to write the trait on the side. So big T, little t, big T, little t. And in each corner, we will write the corresponding two letters from that row and column. So for example, in this first corner, because the row is a capital T and the column is also a capital T, we will write two capital T's. If it is homozygous dominant, the dominant phenotype will show. If it is a homozygous recessive, the recessive trait will show. However, if it's heterozygous, the dominant phenotype will show because the dominant allele basically trumps the lowercase or recessive allele. So that's why in this monohybrid cross, we will have 3 out of 4 offspring will generally be tall, whereas there will be one short plant out of every 4. A test cross helps determine the genotype of a parent given both phenotypes and a population of its offspring. So in this example, if we know that we have a parent that is purple and a parent that is white, and we know that the dominant allele is capital P and that means it's purple, whereas a lowercase p or the recessive allele will make it white, and we also observe the population to have all purple plants, then we will know that the parent will have a homozygous dominant for big P. However, if we see that about 50% of the offspring are purple, then we know that the parent will have to be heterozygous in order to receive this monohybrid cross. A dihybrid cross will involve the mixture of two separate genes. So in this case, a capital R will denote that it's round, a lowercase r will mean it's wrinkled, and that's the recessive trait. And for color, a capital Y means it's yellow, and a lowercase y means it's green. So in this cross, both parents will be heterozygous for both traits. And in order to figure out what we put on the top and the side, we will use FOIL, or first, outer, inner, and last. So for example, if it's First, we will use capital R and capital Y. However, if it's outer, we will take the outer two letters. So we will take a capital R and a lowercase y. For the inner, we will take a lowercase r and a capital Y. And finally, for the last, we will use the lowercase r and the lowercase y. And we have written those on the top and the side. And once we combine all the letters together, we will see that we will come to a phenotype ratio of 9 that are round in yellow, 3 that are round in green, 3 that are wrinkled in yellow, and 1 that is wrinkled in green. An X-linked cross trait or a sex-linked trait is when the gene in question is found on the 23rd chromosome, or the one that determines the gender. Males will always have an X and a Y, and females are always X and an X for their 2 chromosomes. And hemophilia is an X-linked recessive trait. And this diagram will show a father that is unaffected because hemophilia is recessive, whereas a mother will carry the trait and being heterozygous. And from this cross, we will see that the dominant allele will always show if it's present. However, if there's a recessive allele available, the trait will show, unless the dominant allele trumps it. Now let's talk about the many types of non-Mendelian genetics, or exceptions to the standard cross. Incomplete dominance is when the traits blend together. So in this case, when a homozygous red plant mixes with a homozygous white plant, we will get a capital R and a lowercase r. And all of those plants, rather than being either red or white, they will become a mixture of them, or pink. Codominance is when both traits will actually show. So in this example, a white cow mixed with a red cow will result in a cow that is a mixture of both colors. Epistasis is when the genes controlling one phenotype will affect another gene at a different location, and it can mask a trait. So for example, when you do a normal cross, you might get a blonde hair or a red-haired person. However, if that person has a gene for baldness, then they will become bald no matter what the gene for their hair color was. Pleiotropy is when one gene controls many phenotypes. So in this situation, this gene over here can affect 3 different traits. Polygenic inheritance is when many genes for one phenotype will add together to contribute to a range of different phenotypes. So in this example, we use skin color. Multiple alleles is when there is more than 2 alleles that control a trait. So this is prominent in the blood types. So if you have a blood type of O, you will always have a genotype of 2 lowercase i's. However, if you have a blood type of AB, you will have a genotype of A and B. If you have a blood type phenotype of B, you can either be homozygous for the B, or it can be heterozygous using the lowercase I. And the same goes with the blood type of A, except the genotype, you will use A instead of a B. Next are sex-linked traits, which we talked about in the previous slide. And these are genes located on sex chromosomes. Finally, environmental effects. So an Arctic wolf will change color depending on its environment or the season. Now let's talk about pedigrees, which show the ancestry of an individual. Usually in pedigrees, a circle will denote that someone is a female, whereas a square will mean that they are a male. And if they are affected with a trait, it will be denoted with a shaded color. So in autosomal dominant pedigrees, they will appear in all generations. And autosomal basically means it's not sex-linked. In autosomal recessive, it can skip generations. In an X-linked dominant pedigree, if the father has it, the daughter must have it. Finally, in X-linked recessive, it will usually affect males more because they only need one recessive trait instead of 2 in order to have the trait. Because a male will be XY and it only needs one X with the gene. Next, let's talk about genetic linkage. Unlinked genes are when genes are on different chromosomes or are far away on the same chromosomes. And when this happens, independent assortment will occur, and the allele received for one trait will not affect the allele Now let's talk about genetic linkage. When genes are considered unlinked, genes are on different chromosomes or are far away on the same chromosome. When this happens, independent assortment occurs, and the allele received for one trait will not affect the other gene. And this is because during crossing over, the exchange of DNA fragments containing one gene will not affect the other, and there is a high likelihood for recombinant configurations of alleles. So you can see in this diagram crossing over, you can see that the two genes are far away. So when they cross over, they will not combine together, and as a result, there is an equal likelihood for each of the gametes that are possible. However, if the genes are considered linked, they are close together on the chromosome, and they are more likely to stay together during crossing over. And if a person has an allele for one trait, they will likely have the allele for a trait with a closely located gene. And during genetic linkage, recombinant configurations are less likely, whereas parental configurations of alleles are more likely for an offspring. And a parental configuration is a pair of alleles that match that of their parents. So in this situation, you will find that an offspring is much more likely to have a parental gamete rather than a recombinant gamete. And recombination frequency is a method used to determine if genes are linked. So here are some basic terminology. The P generation is the parental generation, and it will be used in step 1 when we go over it. The F1 generation is the first generation and is the offspring of the parents. And the F2 generation is the second generation, or the offspring of the offspring. In this problem, the purple gene will be denoted with a PR plus being dominant, meaning it will have red eyes, and a PR being recessive, or purple eyes. And there will be a second gene called the vestigial gene, and VG plus is dominant, meaning it will have long wings, whereas a VG without the plus will be recessive and will denote short wings. So the first step is to breed a homozygous dominant for both traits and a homozygous recessive for both traits in order to guarantee that we get an individual that will be heterozygous for both traits. Using this heterozygous offspring in question, we will breed that with a tester, and this tester will be homozygous recessive for both traits. If the genes are unlinked, the 4 different phenotypes will show up with equal frequency. However, if the genes are linked, the parental phenotypes will show up with greater frequency. And finally, to find the recombination frequency, we will do the recombinants divided by the total offspring times 100%. And in this case, our recombination frequency will be 10.7%. This means that the genes are likely to be linked. Linkage maps are chromosomal maps based on recombination frequencies. So in a linkage map, we will start with the largest crossover frequency, and this number will usually max out at 50% because beyond that, the genes will be unlinked. And the percentage will indicate something called map units. So first we will see that 45% will be the largest. So we will put the B and the C on opposite sides, and 45 map units will separate them. The next step, we will see that B and D is 40%. And since B will likely not go the other way, we will put it 40 map units to the right. Next, we will consider the AC. And the crossover frequency for that is 30%, so we will go backwards from C to A, keeping the B to D. And finally, to check our work, we can find that this D to C will be 5%, because 40 plus 5 will be the total 45. And because this is 5, we will find that AD will have to be 25, and that matches up with what we originally had in the question. The Chi-square test will determine whether or not the results deny or confirm a certain question. So in this question, it states that in the garden pea, the yellow color is dominant to green, and the inflated pod shape is dominant to the constricted form. Considering both of these traits jointly in the self-fertilized dihybrids, the progeny appears in the following numbers. And the question asks, do these genes assort independently? So the first step is to develop a null hypothesis, and this is when there is no statistical significant difference. So in our example, we will say there is no statistically significant difference between the expected offspring population and the observed offspring populations, which would mean that the genes are unlinked. The alternative hypothesis for this, if we disprove the null hypothesis, is there would be a statistically significant difference, and the genes would be linked. The next step is to determine the expected results through a ratio in the dihybrid cross. And because of the 9 to 3 to 3 to 1, when we cross two heterozygous plants, and there should be 994 total offspring, we are able to find the observed and expected. And the observed is what we had in the question, and the expected is found by multiplying the percentage by the total number of plants. In the next step, we will use the equation to find the chi-squared value. So in order to find the chi-squared value, we will find the sum of the observed minus the expected squared over the expected. So let's just take 1, for example. 556 would be the observed number of yellow and inflated plants, while 559 would be the number that we expected, and we divide that by 559. And we would sum that for the green inflated, the yellow constricted, as well as the yellow inflated and green constricted. And when we add all of those numbers up, we get a value of 0.312. In step 4, we use the chart of critical values and use the degrees of freedom of n minus 1. And n will be the number of different phenotypes, and a p-value will be 0.05. So there are 4 different phenotypes as shown in the question, so we subtract 1, and the degrees of freedom will be 3. And we use a p-value of 0.05. And we get a critical value of 7.815. If the chi-squared value that we've previously found was less than the critical value, it is within the limits of chance, so we do not reject the null hypothesis. Otherwise, we will reject the null hypothesis. But in this question, since the calculated value is much lower than the p-value from the table, we cannot reject the null hypothesis. The genes will sort independently according to the 9 to 3 to 3 to 1 ratio and are not on the same chromosome. We can also use the chi-square test in Hardy-Weinberg equations. The first Hardy-Weinberg equation is p squared plus 2pq plus q squared is equal to 1. And p squared is the dominant homozygous frequency within a population, whereas 2pq is the heterozygous frequency within a population, and q squared is the recessive homozygous frequency in a population. And we relate p and q by p plus q is equal to 1. And p is the dominant allele frequency within the entire population, and q is a recessive frequency. So in this question, in a field of flowers, there were 3 phenotypic colors for a particular species, for purple, violet, and white. So this would be an example of incomplete dominance. It was determined that the color was controlled by a single autosomal gene that exhibited partial dominance. The following counts were made. The first step is to find the allelic frequencies. So in order to find p, we will multiply 243 times 2, and we will add 440. We will multiply 243 times 2 because that is homozygous capital P, whereas we will only add 1 times 440 because heterozygous violet flowers will only have 1 P. On the other hand, the P prime, we will multiply 36 times 2, add 440 to get a total of 512 alleles. In order to find the frequency, we'll divide that by the total number of alleles available, which is 1,438, and we will get a P frequency of 0.64 and a P prime frequency of 0.36. The next step is to state our null hypothesis, and our null hypothesis is that the flowers are in Hardy-Weinberg equilibrium. Next, we will apply the equation in order to find the expected counts. So the Hardy-Weinberg equation would be p squared plus 2pq plus q squared is equal to 1. And using the frequencies that we found, we determined that we will get a 0.4096 plus 0.4608 plus 0.1298. In order to find the total number of expected flowers, we will multiply the ratio of the number of flowers to the total number of flowers. And we will get 295 homozygous purple, 331 violet, and 93 white flowers. Next, we will use the chi-squared equation, and we will get a value of 79.89. And finally, because 79.9 is greater than 5.99, which is the critical value that we found with 2 degrees of freedom and a p of 0.05, we will reject the null hypothesis, and it is not in Hardy-Weinberg equilibrium. Thanks for watching. This lesson covered Mendelian genetics, non-Mendelian genetics, pedigrees, linkage and recombination, the chi-square test, and Hardy-Weinberg equilibrium.